Hi folks, this is Dr. V. Sivaramakrishnan from SRM Institute of Science and Technology. I welcome you all for this presentation on genetic disorders of sex chromosomes. These sex chromosomes are responsible for determining the biological sex of an individual. If you consider in the case of females, there are two X chromosomes, and in the case of male, there is one X and one Y chromosome. When there are variations in these sex chromosomes beyond the normal 2x and as well as the XY pattern, then invariably it results in actually the sex chromosomal abnormalities. The abnormalities can happen due to an presence of extra chromosome, or it could be a missing chromosome, or it could be even an altered chromosome. What could be the cause of such an abnormality? The variations can arise whenever there appears an error during the cell division process due to a mechanism which is called as non-disjunction of the chromosomes. And these abnormalities invariably can cause numerous structural changes and also can lead to the abnormalities resulting in a variety of medical, developmental, and as well as psychological conditions. What kind of conditions wherein we can associate? For few examples I can state the syndromes like the Turner syndrome, wherein there is only a 45 chromosomes and an X chromosome where the one of the other chromosome is basically missing, and whereas in the case of the Klinefelter syndrome, whereas there is only 47 chromosomes with XXY, wherein an extra chromosome of an X is basically present. So what ways you can actually identify these sex chromosomal abnormalities? There are currently two possible techniques which basically enable to identify it at a very early stage. That involves either using the karyotyping procedures or an advanced technology which is called as fluorescence in situ hybridization. Having understood the sex chromosome and their role, it is also important, in addition to what I just now mentioned about the two experimental techniques of karyotyping and FISH, which are critical for the postnatal kind of a diagnosis. How about if you want to still more earlier to find the occurrence of such a syndrome, can be also possible using a technique which is called as amniocentesis, and also we have the chorionic villus sampling based approach. But these are prenatal diagnostic procedures which help to also diagnose the occurrence of these syndromes. In addition to this, there are also sequencing methods which are also available, which helps in actually performing the genetic testing, which is essential for diagnosing such disorders that are related to sex chromosomal abnormalities. Now, once if we have identified such occurrence of sexual abnormalities, how could they be managed? Invariably is possibly due to the based on the type and severity, and as well as each one there is no magic bullet, so invariably you required a tailored approach for the treatment for each of these conditions. Now let us explore the first sex chromosomal abnormality, which is called as the Turner syndrome. And this is a genetic condition that affects females. It is caused by absence or a partial absence of one of the X chromosomes. This can be due to complete loss of one X chromosome, or it involves the occurrence of mosaicism, wherein some cells have a normal double X chromosome pattern and the others have a 45X pattern. Turner syndrome is typically diagnosed through the karyotyping approach, wherein a blood test is basically performed to examine the number of chromosomes present in them. And as I mentioned just now earlier, we have both the prenatal and as well as the postnatal diagnostic approaches which can enable to identify the occurrence of Turner syndrome in a female. Now let us see the second genetic condition which occurs particularly in the case of males. It is called as the Klinefelter syndrome. It is caused by an extra X chromosome resulting in 47 chromosomes in total. What could be the characteristics? Individuals with this condition, they have a karyotype of 47XXY, indicating two chromosomes of X and one Y chromosome. This occurs due to the one of the random genetic events that happens during the cell division in the case of again sperm, and the incidence is seen 1 in 500 or 1 in 1000 male births and is often diagnosed in the case of adolescence or at the time of adulthood. Common symptoms include a taller stature, delayed puberty, and infertility, among others. Invariably, they have broader hips, longer legs, and larger breast, weaker bones, a lower energy level, smaller reproductive genitals, and as well as a delay in puberty, as I earlier mentioned, and less facial and as well as body hair in these people. The next genetic condition is called as the triple X syndrome. This is a genetic condition which is often affecting the females, which is characterized by an extra X chromosome. It is also called as the super female syndrome due to the presence of an additional X chromosome. The karyotype for the triple X syndrome is 47 triple X, indicating it has 3 chromosomes present, and individuals with this triple X syndrome, they have a total of 47 chromosomes instead of the standard 46. The extra chromosome generally leads to mild symptoms and it does not cause significant health issues. However, it is often said to be undiagnosed due to its mild nature and lack of noticeable symptoms. The individuals are said to be having an above average height and they have delayed speech development, they have mild intellectual disabilities, menstrual irregularities, and as well as premature ovarian failure. The last genetic syndrome which is associated with the sex chromosomal abnormalities is called as the XYY syndrome, or also known as the Jacob syndrome. Here too, there is an additional Y chromosome present, and due to which they are represented like 47XYY syndrome, it is called as. So this is a genetic condition which is specifically affecting the males, and it is caused due to a random error during the sperm cell division. An extra Y chromosome is present in the fertilized egg due to this particular kind of error, and this occurs in approximately 1 in every 1000 male births. While many with XYY syndrome are said to be healthy, most of them experience behavioral issues and as well as developmental delays. Diagnosis is typically made through karyotyping in identifying the extra Y chromosome, and the management basically involves providing them with educational support and as well as possible hormonal therapies for overcoming the developmental challenges. Now let us see the aspect of understanding about karyotyping as one of the critical techniques that is important for diagnosis of sex chromosomal abnormalities. It's a definitive method for diagnosing most of the sex chromosomal abnormalities, allowing for a precise chromosome analysis. Towards the left, I have represented here the image wherein it involves either collecting samples from the blood or maybe skin or maybe even amniotic fluid. This is basically cultured and by adding phytohemagglutinin, and once these are cultured at 37 degrees centigrade for 3 days, and then they are actually the metaphase chromosome stage at which where they are actually blocked at is performed due to the addition of a chemical which is called as colchicine and an addition of hypotonic saline. And when the cells are basically fixed after being spread onto a particular kind of a slide, invariably digesting it with trypsin and also staining with the dye which is called as GIMSA, which is actually a basic dye which interacts with the phosphate negative charge groups of the DNA which is present or in the chromatin or the chromosomes components, invariably results in forming a G padding kind of a pattern. This enables for the usage of softwares to actually now organize the chromosomes based on their size and as well as based on their composition, invariably from one first chromosome until the sex chromosomes, invariably all the 23 pairs. This is an important technique which provides a critical insights into the genetic disorders which are linked to the sex chromosomal abnormalities, aiding in an early diagnosis. We have seen the importance of a postnatal diagnostic technique. Now let us see the importance of a prenatal diagnostic technique, which is called as the chorionic village sampling. This is a prenatal test for detecting the genetic abnormalities as early as in case of pregnancy, and it is conducted anywhere between the 10 and 13 weeks of gestation, making it an early diagnostic option. How is this basically performed? A sample of the placental tissue, particularly the chorionic villi component, is collected using a needle or a catheter through the cervix or abdomen as shown in the figure. And the purpose is it involves the analyzing the fetal chromosomes by this approach for occurrence of any genetic disorders or abnormalities much earlier than before the birth. And tissue samples are tested using further in the case of the postnatal diagnosis after birth by using the karyotyping or the fish approaches, also for identifying the sex chromosomal abnormalities. Now, there is one small risk that is basically associated with this particular kind of CVS approach or the chorionic village sampling. While it is considered to be generally safe, it also carries risks such as miscarriage, infection, and as well as bleeding that can basically happen with this, because this is actually an invasive procedure. And early detection through the CVS allows for informed decision making regarding the pregnancy management, and hence this is basically considered as one of an important technique for even genetic counseling also. I thank everyone for your kind attention and as well as your patience. I appreciate your time and as well as interest today in this particular topic. Thank you.